A warm welcome to botany class. In the previous class, we discussed asexual reproduction in plants, in the lower form of the plants. Today, we are going to discuss sexual reproduction in plants. Sexual reproduction in plants. Earlier, you learned about asexual reproduction with several examples, mainly in the lower forms like algae as well as bryophytes. Let's start this class with sexual reproduction in higher plants. Before that, let us see how sexual reproduction takes place in algae and bryophytes. Algae and bryophytes, as you know, it's the lower form of organisms. Normally, this sexual reproduction undergo two main processes, namely the production and fusion of gametes. The production of gamete, the process is called as gametogenesis, and the fusion is called as fertilization. There are two processes under which gametogenesis and fertilization. What's meant by gametogenesis? The production of gametes. The male as well as female gametes are formed by the process called gametogenesis, and fertilization is the process of fusion of male and female gametes. So, these processes are included under sexual reproduction. Let's see how this reproduction, this gametogenesis, how this reproduction taking place in algae and bryophytes. There are three different stages in the gametic fusion named as isogamy, anisogamy and oogamy. Isogamy, anisogamy and oogamy. What's this isogamy? The meaning of iso means same or similar. So, in this fusion, isogametic fusion, the gametes are fused. The gametes or the nature of the gametes are morphologically and physiologically similar gametes. As the name indicates, iso, similar gametic fusion. How it is similar? Morphologically and physiologically the gametes are similar. That is named as isogametic fusion. Then anisogamy. Anisogamy, the gametes are either morphologically or physiologically dissimilar. Dissimilar gametes either morphologically or physiologically is called anisogamy. Then another type is oogamy. Both morphologically and physiologically the gametes are dissimilar. So, there are three different types: isogamy, anisogamy, oogamy. Isogamy or similar gametes, anisogamy either physiologically or morphologically dissimilar gametes, oogamy both the gametes are dissimilar, completely it is dissimilar, morphologically as well as physiologically. That type is oogamy. And flower is very important. You all know about the parts of the flower, because flower is the reproductive organ of the plant. So, this flower, already you learned earlier the parts of the flower, which consist of four different whorls named as calyx, corolla, Androecium and Gynoecium. Everybody admire flower. Flower. What is meant by flower? Flower is the inspirational tool of the poets, and the flower, which is the decorative material. In all the celebrations, we used to decorate with the flowers. In Tamil literature, the 5 different lands are denoted with different kinds of flower, and in some countries, the flags are embedded with flowers. But morphologists, they said that the flower is a condensed, highly condensed shoot. For morphologists, a flower is a highly condensed shoot meant for reproduction. So, morphologists explain the flower is a highly condensed shoot meant for reproduction. And there are different worlds of flower. As you learned earlier, there are 4 different worlds. The first world or the outermost world is called as calyx. This is the outermost world of the flower, which is named as calyx. And the second world of the flower, this world, second world is corolla. Each corolla, this world, second world is named as corolla, and each segment of this corolla is named as petal. Before that, the calyx, which contains number of whorls, number of units. So, each segment of this calyx is named as sepal. Each segment of the second whorl, this is the second whorl of the flower. The whole whorl is corolla, and it is made up of number of segments. Each segment of the corolla is called as petal and androecium. So, androecium and gynoecium. Among these 4 whorls, this androecium, which is made up of stamen, and the gynoecium. Today we are going to discuss in detail about androecium. Androecium, you know, it is the male reproductive part. So, this androecium, which is composed of, or it is made up of stamens. This is the bisexual flower. Now, this part is the androecium, which is composed of number of stamens. So, this stamen, which consists of anther, stamen consists of anther and filament. The stamen is made up of, this is the stamen which is made up of anther and filament. Anther and filament. Inner to this anther, the pollen grains are present. These are the pollen grains. The pollen grains which is represent as the male gametophyte. The pollen grains are represent as the male gametophyte. Now, today we are going to discuss in detail about the development of anther. Here there are some diagrams which shows the development of anther. So, this anther, how the anther developed? When the anther is young, the young anther developed as a homogeneous mass of tissue. The young anther develops as a homogeneous is the similar kind of cells or same kind of cells. So, this is the homozygous mass of tissue which is similar cells, homozygous cells present inner to the epidermis. The outermost layer is called as epidermis. Inner to the epidermis, there are number of homogeneous mass of cells. In young, it developed in this manner. Now, this anther, which looks like, it's assumed as 4-lobed structure, which looks like a 4-lobed structure. here in the initial stage, which assumes as a four-lobed structure. Inner to the epidermis, you know that outermost layer of each and every part is made up of epidermis. Inner to epidermis, there are some layers of hypodermis. Here in the next stage, first it is formed, the young anther forms as a homogeneous mass of tissue, and this hypodermal cells, inner to that there are some hypodermal cells. These hypodermal cells, a row or a few cells are enlarged with conspicuous nucleus. The cell of hypodermis enlarged with conspicuous nucleus, and it becomes an initial called archisporial cell. So this cell functions as a archisporial cell. So first stage is the homogeneous mass of cells or tissue. Then from the hypodermal cells, there are few rows of cells or few cells enlarged with conspicuous nucleus, and it becomes archisporial cell. Now these are the archisporial cells. These archisporial cells undergo divisions. Mainly there are two kinds of division. They are periclinal division and anticlinal division. There are two types of division, periclinal and anticlinal. What are the difference? The periclinal division, which is parallel to each other of the tissue. So these all cells are parallel division. It divides like this, parallel to the tissue, periclinal. But anticlinal, it divides like this type of division, anticlinal division. So this is periclinal division, this is anticlinal division. So both the direction the cells divides. Now this archisporial cells undergo periclinal division, and it forms two different tissues. They are primary parietal tissue and primary sporogenous cell. How these cells are formed? Primary parietal cell and primary sporogenous cell. The archisporial cell divides periclinally and forms two different kind of tissue. The tissue which is present towards the peripheral region, as the name is, a primary parietal tissue. Parietal tissue which occurs in the peripheral region, towards the periphery. And inner to this, inner cells, they are formed as the primary sporogenous cells. Now from the archisporial cell, there are two different types of tissues are formed. They are primary parietal cell and primary sporogenous cell. Again, this primary parietal cell, now this primary parietal cell undergo divisions. So when it undergo divisions, periclinal and anticlinal divisions, which produces 2 to 4 layers of, there are 2 to 4 layers of anther wall. Look at this diagram. Here there are different walls. These walls are composed of endothecium. The outermost layer is epidermis. Now it forms endothecium. Endothecium. Inner to endothecium, it forms middle layers, tapetum, and so this layer endothecium, middle layers and tapetum is formed. 2 to 4 layers are formed from the primary parietal cell. From primary parietal cell, the wall layers are formed. What are the wall layers? The endothecium. Here this is the epidermis. Inner to this layer, here this layer is middle layer, and inner to this tapetum. Inner to epidermis there are endothecium. Now this sporogenous cell, primary sporogenous cell undergoes mitotic division. It undergoes mitotic division and it forms a mass of sporogenous tissue. So in this diagram you found that there is a sporogenous tissue. The wall layers are surrounded by the sporogenous tissue is surrounded by the wall layers. So, how this sporogenous tissue formed? This is the sporogenous cell which is derived from primary sporogenous cell. The primary sporogenous cell undergo mitotic division. One word question, these are important. The primary sporogenous cell undergo mitotic division and it becomes sporogenous tissue. Now, in this anther, so here one part only enlarged. Here it contains epidermis. The outer layer is the epidermal layer, this layer is middle layer and this layer is tapetum. In the mature anther, it looks like the outermost layer is epidermis, this is the epidermal layer, outermost layer. Inner to this, there are endothecium. This layer is endothecium. Inner to endothecium, there are middle layers. There are 2 to 3 layers of middle layer. And inner to this middle layer, there is tapetum. This is tapetum. So, this is epidermis. Outermost layer is known as epidermis. This layer is endothecium. This layer is middle layer. And this is tapetum. These are the wall layers. In the mature anther, you may see this wall layers in detail. Next class, you are going to study detailed structure of this mature anther. Now, these all wall layers are developed. Now, the microsporogenesis, the next process is known as microsporogenesis. What is meant by microsporogenesis? It's the process by which formation of microspore. Microspore, it is haploid. The microspore is haploid, which is represented as N. Microspore. How it is formed? The microspore is formed by meiotic division. Meiotic division of microspore mother cell. How this microspore mother cell forms? From this primary sporogenous cell, mitotic division takes place. So, it becomes sporogenous tissue. Now, the last generation of sporogenous tissue, there are number of stages. The last generation of sporogenous tissue become microspore mother cell. Now, this microspore mother cell is diploid, 2N. Microspore mother cell is diploid, 2N. Microspore mother cell undergo meiotic division. When it undergo meiotic division, it becomes microspore tetrad. So, now it forms as a microspore tetrad. 4 haploid microspores are formed. So, at first, in the first stage, microspore tetrads are formed. Here, there are 4 different haploid microspores are there. They may be arranged in linear. There are 4 cells, 1, 2, 3 and 4. Linear, either it is arranged linear or decussate in this form, or isobilateral, or several, or it is arranged in T-shaped. This is the structure of T-shaped. The tetrahedral microscope arranged in different shapes. Now this microspore tetrad, from the microspore tetrad, each microspore is derived. So each microspore is developed in the final stage, and the microspores remains located in each lobe and becomes the pollen grain. Now it becomes the pollen grain. So in this way the anther developed. So pollen grains are formed. I hope that you understand very clear. So these all diagrams are very important. You practice all these diagrams in the class note. You do not have the book now, so you follow these diagrams. This is the first stage, and how this anthers are developed, anther wall is developed, and how this microspore tetrad is formed. Finally, from the microspore tetrad, microspore and pollen grains are developed. Learn all these questions before the next class. Thank you.